All right, welcome to week 15. Welcome to our last full week of lectures and work. This is our final chapter we are going to cover, which is chapter 28, the reproduction system. For the final exam, there may end up being a few, I think there's like 3 questions maybe from chapter 29 related to egg and sperm interactions, meiosis, and fertilization, and journey to the uterus and implanting. But for the most part, other than the final exam that's showing up, this is our last chapter of content on our lecture test, on our lab exam. And again, most of you can probably potentially answer those questions, educated guess those questions, and still be fine for the final exam. All right. Okay, so again, our reproduction system, this is the one system that if you remove, you can actually live without. All right, it starts off in an embryo as the same, and then with the Y chromosome, and if the Y chromosome is functional, it will then generate the signaling markers, the signaling molecules to push differentiation from a genetic female phenotype to the male. All right. Now, is there a possibility, and you hear about this sometime, that some of us phenotypes walking around that are female, so we currently are walking around as females, we might be genetically speaking males, we just had a faulty Y chromosome that didn't produce the molecules to push the maturation and differentiation into the male phenotype. That is possible. Okay, so again, you might do this in a little genetic thing where you take some cells from your body and you crush them up, and then you look for, do I have 2 XX barbells, or do I have just one? And so would I be potentially a female genotype, female phenotype, or am I a female phenotype but actually a male genotype? Okay, again, it's for genetics, and again, some of that information you might be able to get into if you take a bio class with Dr. Peters or Dr. Harms in the Bio 140, and then the genetics heredity classes. Okay, they go through more of this. All right, and then you might get some more of this information if you take Jody's biology, the sex biology class about reproduction. All right, so we have 2 versions of this system. There's a male side and a female side. And again, some of the textbooks will say we have 12 organ systems, male and female are 2 separate. And then other textbooks say we have 11 organ systems, and the 11th is divided into A, which is female, or B, which is male. So it's just semantics, but we have male and female versions of this organ system. All right, and the purpose of this organ system is to make gametes, so make haploid cells. In the male, that is known as a sperm cell. In the female, that is known as a egg cell, the oocyte. All right, and then that's make them. Then you have to try to bring them together. Okay, and the reproductive tract is going to be all the chambers and passageways that are trying to connect to the male and female when they come together to try to bring those cells towards each other. Okay, now when we try to refer to things in a non-gender way, we are going to call our reproductive organs the gonads. Okay, so in the male, the gonads are the testes. In the female, the gonads are the ovaries. So in the testes, that is where you take a cell, you make it go through meiosis, so it goes through 2 divisions and gives you 4 sperm cells, right? In the female, the ovary is going to have a cell surrounded by a bunch of helper cells. That one cell is going to, under the right conditions, go through those 2 divisions, but it's only going to produce 1 egg cell and 3 polar bodies. And the 1 egg cell is what the LH surge is going to try to ovulate out and then get towards and close to those sperm so you can get fertilization. Okay, now there is going to be lots of tubing, lots of glands, lots of passageways that are going to try to support and give fluid and liquid to keep the sperm and the egg cells suspended in. and provide them nutrients, as well as there's going to be tubing and passageways for those molecules to make their way towards each other. Okay? The external genitalia, and again, in the male, that's a pretty pronounced penis. All right. In the female, it's going to be the labia majora, minora, the mons pubis, the clitoris, the hymen. All right. It's not quite as visible, but it's still that area that's externally located, pieces and parts of the body that are going to, again, play a role in bringing the gametes towards each other. And you can collectively call that the external genitalia. Okay? All right. So the male and female systems are a little different. The male is all about producing lots and lots and lots of gametes and then delivery. The female is about putting its egg and putting its investment into one viable option a month, and then housing that viable option, if it gets fertilized, implants and can develop and grow until it's at a stage that the offspring can come out and somewhat survive on its own. Okay, so the female has two parts in that it has to grow its gamete, and then it has to get the gamete to fertilize and then grow the embryo, grow the new entity until the new entity has the ability to be viable outside of mom's protection and body. Okay, now when we think about other animals in the animal kingdom, when the female expounds like a horse or a zebra or, you know, little antelope, those little guys, they need to be up and running and walking within minutes to hours of their birth because mom needs them to move with the herd. Our offspring is not, or they are not born mobile. They are not born at that point of mobility. So again, there are some differences in the world of offspring when they leave the mother. Their viability and ability to maybe survive and make it on their own, or with little to lots of support. There's a spectrum on their mother. Okay. All right. Now, when we look at it again, the number-wise, males then can produce up to 500 million sperm per day. So remember, that's about 500,000 sperm, 500 million sperm. All right. And then females are trying to produce one per 28-day cycle. So when you think about the ability to produce offspring, you're trying to find a female producing one viable egg, and then you're trying to get the timing of this egg that it's accessible for the sperm to interact with and potentially fertilize. And then you're still waiting for this now fertilized embryo to continue its journey and implant. So the odds of, getting pregnant and taking that pregnancy through potentially the first trimester, there's a lot of stopping points where a check has to happen that this happened, this happened, this happened for continuation of and maturation of that embryo. Right? And that's why in some cases it seems like it's a miracle that pregnancy can happen. Right? And then as we age, again, in females, you are born with all of the potential viable eggs that you are able to make for your lifetime, and you will start to produce and, again, mature those eggs in a manner each month from about puberty to about 40-ish, 50-ish years. So your window is, again, if puberty happens around 12, and let's say you start going through menopause at 45, 47, it's about, what's 47 minus 12? About 35 years or less of time that you are a female capable of reproduction. Males, you will start producing sperm again in puberty, and males will maybe hit puberty around 13, 14, 15, but they will continue to make sperm for the entirety of their life. Now, there is with age, no, Allie, you don't need that. There is with age, the number of sperm will decrease. And if you look at the data out there, male parents over 50, there is higher incidence and risk of schizophrenia and other things that can happen because, again, your sperm are old and they've been exposed to lots of crap. Especially the cells they come from have been living in your system for a long time and been exposed to lots of crap. So the odds that something is tweaky or not quite as perfectly smooth and oriented, can happen. Okay? All right, so we're going to start with the males. All right, so the male gonads are the testes. You have two of these. They are going to sit slightly outside the body, and they are going to be surrounded by skin. And then just like any kind of organ, it's going to go epidermis, dermis, hypodermis. Then you're going to have muscle. Then you're going to have a little bit of a cavity created that is going to try to, again, let the skin and the muscle have a little freedom of movement from the egg of the testes, the little pouch of the testes. All right. In the testes, you are going to have cells that are going to go through meiosis and become sperm. You're going to have cells that are going to support and nurture and provide input and influence through cytokines and paracrine factors. Those are going to be your Sertoli or your nerve cells. So those two cells are going to live in little tubing known as your seminiferous tubes. Okay? The interstitial area is, of course, going to have connective tissue cells like fibroblasts, fibrocytes. It's going to have endothelial cells. It's going to have blood vessels. And then living in that connective tissue is going to be a new population of cells known as Leydig or interstitial cells. Those are going to be the cells that are going to produce the testosterone. So they are going to take cholesterol being something like 30-something molecule carbons and those 4-ring structures, and they are going to convert cholesterol to the steroid hormone androgens, which is collectively testosterone and its many forms. Okay? All right. The female side, the ovaries are going to be the paired organs that have the egg cells, the cell that is going to go through meiosis, and that egg cell is going to be surrounded by the supporting cells. And those supporting cells are going to grow with maturation of the unit being the egg and the supporting cells or a follicle. All right. So when a female is born, she has all of her follicles, and she may have 1 million follicles in the right ovary and 1 million follicles in the left ovary, and that's it. In those follicles are one little cell that can go through meiosis to potentially provide one viable egg cell. And once you go through puberty, every month you're going to push a few of those follicles to start to develop. So of your 1 million follicles on the right and 1 million follicles on the left, it might be that you only end up with 250 actual viable eggs in your lifetime from each ovary. All right, and that you have a lot of attrition. Okay, all right. So the ovaries are going to be suspended inside the body, and they are going to be suspended not on midline. What am I trying to say? In that plane, and then they will have they'll be wrapped around by some little like finger-like tubing that is going to, when an ovum or an egg is released by ovulation, try to capture the ovum so it can go into the tubes that then connect to the on midline uterus, where that, if the egg is fertilized, could implant and continue to grow and mature until it's viable enough to be pushed out. Okay? So the uterine tubes are sometimes also known as the fallopian tubes. I'm working. I need to work. Please just give me a minute, okay? Go to your library at 12 o'clock. Okay? All right, and then the uterus, the external genitalia in the female is going to be the vulva, which is going to include the vagina and the mons pubis and all the features of the openings and entry points for our ureter, our vagina, and our anus. Okay. I'm sorry, the kids are very distracting. All right, so we're going to focus first on the males. So again, on Thursday, we're going to try to go through some of these key pieces and parts of the male reproductive internal, external features on the models. All right, so again, our males in the testes have this little network of tubing, and inside the tubing are going to be nurse cells and these cells that are going to go through meiosis and become sperm. Okay, so those cells that are going to give rise to sperm, we're going to call spermatozoa. They are going to be inside here, inside the little testes in some tubing. All right, and that tubing is going to then connect in the superior or top through some ducting, through some tubes that are going to eventually connect to an organ that sits on top. That's the epididymis. All right. Then the epididymis is going to need to connect into a tube that gets up and over and around the bladder. And that big tube that goes up, over, and around is your ductus deferens or your vas deferens. And your males who get a vasectomy, this is where they're trying to cut. They are trying to go right here in the scrotum sac. They're going to try to make a little incision, and they're going to try to cut the tubing so the epididymis is no longer connected to the ductus deferens, and then any sperm created would start to just exit into the interstitial space, the hypodermis space, and be lost to any kind of immune cells that are living in that connective tissue environment. Okay. Now, when you are sitting in the vas deferens, there's a little enlargement that kind of holds sperm here. All right. The vas deferens needs to get into the prostate gland and connect to the urethra. All right. So the connection from here to the urethra through the prostate, the tubing, the doorway, or the roadway at this point, is known as your ejaculatory duct. And as things go into the ejaculatory duct, secretions from the seminal vesicle, which is sitting right here next to the vas deferens on the back of the bladder, and the prostate are going to join any sperm as it then pushes in past the ejaculatory doorway, ejaculatory duct, into the urethra. So what gets into the urethra is going to be sperm, secretions from the seminal vessel, and secretions from the prostate. And those three things together are now going to be your ejaculate. Okay? Now, the urethra, as you remember from last lab, when it's in the prostate, it's known as the prostate urethra. When it goes through the little muscle layer and the bulbourethral gland, or the Cowper's gland, which secretes preemptively through the urethra to get rid of any acid, liquid, leftover urine and neutralize it, all right? That's that part of the urethra there. Not the prostate urethra, but the, I'm totally blanking on it. And then you have, once it gets into the spongy, surrounded by this cavernosum and the spongiosum, the spongy urethra, that is going to be the three parts of the urethra, and then the exiting point. Membraneous urethra. Thank you. Thank you. See, I'm brain farting. I'm too distracted by the children. All right. So there are three key glands now we've hit on in the male. So the one that's lined up in parallel to the vas deferens near that entry point into the prostate, that is going to be your seminal vesicle. And you're going to have two of those because you have two vas deferens, so there's two seminal vesicles. Okay, this is going to be a large portion of the fluid that's going to join the sperm on ejaculation. That's going to give it some fuel. That's going to give some base to the pH. And it's going to be full of, again, supporting molecules for triggering the uterus to contract, for helping to plug up the openings to try to keep things in the uterus, and to try to, again, protect the sperm. All right. The prostate is going to secrete a little bit of acid, so the acidity and the base kind of like neutralize each other, so I don't get too alkaline and kill my sperm because I made the ejaculate too basic. Okay, so the prostate tries to help me even it out, and again, it gives me more nutrients and fuel and fluid to support the sperm. Okay, the bulbourethral gland, which isn't shown here, but again, it's in that membranous area, all right? It's very, 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 very high in base, so bicarbonate and other bases, to preemptively cleanse the urethra before the ejaculate comes, all right? And so if there's little droplets of urine that have remained in the urethra for whatever reason, it's going to try to neutralize them and then help with lubrication along with some external lubricating glands in the skin of the penis that are going to help, again, to try to lubricate the penis, all right? Because we don't want scratching, tearing, and breaking of the skin there, okay? Now, again, the scrotum. The scrotum is what we call the skin-like sacs that are going to encase the testes, the epididymis, and the beginnings of the vas deferens. All right, they are going to have, again, a mixture of the skin. Looks like it's stretchable, and it has hills and valleys, so it's called Ralphie. The skin is going to have an epidermis layer with some stratified squamous epithelium. It's going to have a dermis layer with mixes of, again, your connective tissue, dense irregular and dense reticular. And then it's going to have a muscle layer that is going to be known as the dartos muscle layer. And the muscle layer here can help, again, move and pull up and down the testes, so the temperature of the testes stays at the optimal temperature for sperm cell development. Okay, there is going to be a little bit of hypodermis, fascia. Then you're going to get into, again, creating a little bit of space of the skin and the muscle can kind of move, and that doesn't necessarily mean if I'm holding an egg that the egg has to be manipulated. All right, so I'm going to create kind of like a little bit of separation between the saran wrap around my steak. Okay, so that is going to be created by the tunica vaginalis and the tunica albugina. All right, and the tunica vaginalis is going to be like the parietal membrane. It's going to be the more close one to the hypodermis, the muscle, and the skin. The tunica albugina is somewhat the like capsule and the connective tissue that forms the outer covering of the testes. And the tunica albugina is also going to cut in and form the testes into little segments that the seminiferous tubules inside can kind of be contained within. Okay. And then the penis is going to be another part of the external genitalia that is for eventually delivery of what is developing in the scrotum, in the testes, inside the scrotum, as it's made its journey, and it's taken maybe 2 weeks to get from original cell to formed sperm cells that are now ready to whip their flagella and swim and try to fertilize an egg. Okay? All right, so again, in this picture, again, I like how it shows the different layers. So again, we have an epidermis, a dermis, you have the muscle, you have the hypodermis layer, and then you're going to have the outer layer that gives you, again, a little bit of separation from, so it's like if this is a shirt, this is the skin and the hypodermis, and the tunica vaginalis is going to be the inner part of this clothing. So I have a little separation, and then this is the tunica albugina, and the tunica albugina is going to push into the testes. And by giving me that, I then, again, create a little bit of freedom of movement between the testes and the skin and the surrounding tissues. All right? And the Ralphie is the raised thickening in the scrotal surface that helps make the 2 partitions that keep your 2 testes separated from each other. Okay. So again, our dartos muscle is going to be a layer of smooth muscle in the dermis of the scrotum. So these are cells in the dermal layer, all right? And they are going to give you some characteristic wrinkling of the skin. Okay, your cremaster muscle is skeletal muscle deeper in the dermis, and this, I'm sorry from earlier, this is the muscle that, based on temperature and reflexive feedback with temperature sensation, is going to pull the testes up or let the testes hang, so the temperature of the testes stays a little bit cooler than that 38 degrees, 37 degrees Celsius, or 98 degrees Fahrenheit. Right? Again, the tunica albuginea is going to be that harder piece of the testes. The tunica vagialis is going to be the separation and the last tissue before you create a little space, and then you hit the tunica albuginea. Okay? And the tunica albuginea is going to push in and create the dissectional chambers in the testes. Okay? All right, so again, in this picture, if we were able to blow it up, you should be able to see the epidermis, dermis, the dartos muscle, hypodermis, cremaster muscle, tunica vagialis, space, tunica albuginea, and the tunica albuginea pushes in. And the tubes are again, tubes containing our spermatozoa and our nurse cells, and they are, again, shriveled up tubes full of cells sitting in these chambers. And as the cells become more mature sperm, they are going to slowly work their way to these connecting doorways that connect to the epidermis. Okay, so how do I connect the testes tubes, the seminiferous tubules, to the epidermis? I am going to have a network of ready testes. That is kind of like the funneling of all these seminiferous tubules towards one area, all right? And the doorways that let you kind of pull them in are the ready testes. So think of it as the major and minor calyces, and the ready testes are similar. And then I'm going to have about 15 to 20 doors that are going to open to the epididymis. Those doors are going to be the efferent ductules. Right? So the ready testes is the atrium, the entryway that you bring all the sperm to sit, and you collect them there, and then you let them physically leave and walk through doorways, the efferent ductules, and then they're in the epididymis. Right? The epididymis, again, is going to have a head, tail, and neck, and it's going to continue to keep the sperm in tubes. Now, back in the testes, again, I've mentioned some specific cells that you have to know. All right, so there are going to be a population of stem cells. These stem cells are cells that, when they divide and go through mitosis, they are going to produce one daughter cell, and then one of the cells that becomes a spermatozoa that's going to go through meiosis. And in the males, you keep these stem cells hopefully for your entire life, which means you keep a small number of these cells that keep going through mitosis, 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 and keep dividing and giving one of the daughter cells to sperm production and the other daughter cell to replacing itself, and that's how you can produce sperm for the rest of your life. Okay, and then when these cells go through meiosis, they are going to go through both divisions. So they're going to divide into haploid, then diploid, or diploid, then haploid, all right, in the testes, and then they're going to start to differentiate as a haploid cell into the head, the neck, and the flagellum. Okay. Now, supporting these cells inside the testicle tubules are going to be nurse or Sertoli cells, right? And these nurse or Sertoli cells function to help guide the number of spermatozoa developing cells to be just right. So FSH is going to be influencing the nurse cells and these mitosis stem cells when and if they go through mitosis, and then when and if so many of them start going through meiosis. Okay, the nurse cells are going to be providing the inhibin hormone to feed back to your brain to make sure your FSH and your sperm counts are just right. So if you have too much FSH, you're going to end up with too much sperm, and when you ejaculate, you're going to end up with sludge that can't swim. All right, so you want FSH to push sperm production. The nurse cells are going to push inhibin, so you don't get too much FSH, so you don't get too much sperm development. So when you ejaculate out, you have a nice sperm liquid balance, so sperm can actually use their flagella and potentially swim. Okay, in the interstitial connective tissue around the seminiferous tubules, you'll see, okay, here's some epithelial tissue, here's a basement membrane, here's a blood vessel, here's connective tissue. There'll be a little fibroblast probably right here laying out this, you know, what do you call it? Fibers, collagen and elastin, all right? There might be some immune cells, maybe some lymphocytes and some macrophages in here, okay? Embedded in this, again, connective tissue are going to be these Leydig cells. Leydig cells are what LH is coming and stimulating these guys to take cholesterol and create our testosterone, all right? And all of our forms of testosterone. Okay, some of the testosterone is going to jump across and help with the nerve cells push against sperm and maturation. Most of the testosterone needs to find its binding protein or albumin and needs to go in the bloodstream and then influence your brain, influence your muscles, influence your blood, influence secondary sex characteristics of our system. Okay. All right, I will not ask you the process of spermatogonia development or spermatogenesis development, but note that, again, there are these cells, and this cell stays in its population, and every few cycles under the influence of FSH, the nurse cells and testosterone will go through and do a cell division, and it'll produce 2 daughter cells. And one of its daughter cells can become eventually a spermatogonia that develops into 4 sperm. And the other daughter takes its place. And then later on, that daughter cell will go through mitosis, produce a daughter to go through spermatogonia, and then the other one replaces. And that's how males, again, should be able to continue to produce sperm for the entirety of their life, as long as these cells do not get destroyed. And when they go through mitosis, one daughter cell remains, the other one goes through the cell division and maturation into sperm. Okay, again, the division is first division, you're still diploid. It's the second division, you become haploid. Now, when you become haploid, if your immune system was to find these cells, they would not recognize them as self because technically they are no longer self. So part of having the tubing of the seminiferous tubules, reticestes, efferent ductules, epididymis, vas deferens, ejaculatory duct, urethra, is the haploid sperm cells. They are always in a tube. They are always protected in a tube from your immune cells, which are in the connective tissue outside the tube. And as long as the connective tissue is where the immune cells remain and they don't ever interact with the sperm cells, your autoimmune system can't start to control or deplete your sperm cells. So, catchers, people who have, this is why you wear a cup in some of your contact sports. If you get hits and you damage and blood and the integrity of your seminiferous tubules, your epididymis, your reticestes, your efferent ductules happens, and immune cells can get in, immune cells are going to wipe out your sperm cells and any cells at the haploid level and below. And they'll make antibodies, and they might create memory cells that live in here and continue to reduce sperm numbers, sperm counts, and reproductive capability. Okay, so that's part of the reason why in many sports you have to wear the cup, is you're doing this, one, because it hurts to get hit in this area, but 2, you're also protecting the integrity of the system and not potentially making immune cells and have access to your sperm and reducing your sperm count later in life. Okay. All right, so again, another picture just to kind of show you how the system works. For the hormones, And again, why the nurse cells and the Leydig cells are so important. Your nurse cells are part of, again, protecting and supporting the push for mitosis versus meiosis and development of those sperm. They do play a role in regulating, this is for sperm count, all right, and giving feedback to the anterior pituitary. And again, the goal is to decrease FSH through negative feedback so I don't overproduce and overstimulate sperm production and make my ejaculate sludge that can't swim. Other things the nurse cells do will, in development, help, again, create the factors that lead to male production of phenotype, not female. And then remember, androgen binding protein, this is so testosterone, which is a steroid, can travel in the bloodstream bound to a protein that helps be water soluble. All right. Just again, this cycle of how inhibin works. So the hypothalamus produces the releasing factor, gonadotropic releasing hormone. It goes to the pituitary gland, and FSH and LH will be secreted. FSH is heading to our Sertoli cells to tell them, and the sperm producing cells in the testes tubing to produce sperm. Inhibin goes and will only try to inhibit FSH levels. So inhibin and FSH are a negative feedback loop. LH is heading to the Leydig cells. The testosterone produced can, again, push in and try to help push sperm cell production, right? But most of the testosterone is going to do secondary sex characteristics and get in the bloodstream. And then as it's traveling in the bloodstream, it can feed back on both LH and FSH and GnRH. So testosterone's negative feedback loop is affecting all of the hormones coming from the pituitary and the hypothalamus. Inhibin only influences FSH levels. Okay. All right, so we've hit most of these things. You have, again, your testes. Your testes push in through the efferent ductules into your epididymis. Your epididymis is going to, again, have a head, a neck, a tail. It is going to be maturation of the spermatozoa, and then the vas deferens take the spermatozoa with peristalsis, so smooth muscle contractions, and ciliated epithelial cells that can help, again, beat and push up against gravity, push over and bring to the back the spermatozoa. Now, one thing to consider, we cannot take a person in a coma and pull sperm out of the epididymis or the testes and then try to fertilize an egg. Let's say the person in the coma is your husband and you want to get sperm from him and fertilize and have children. You can't get sperm from pulling it out with a needle. Sperm has to go through the maturation process and the time spent in the seminiferous tubule, spent in the epididymis, spent in the vas deferens. It has to get the exposure to secretions in the seminal vesicle and the prostate to turn on. So that is why, to get viable sperm capable of fertilizing an oocyte, you have to get sperm from ejaculate. You can't get sperm from an extraction out of the testes or the epididymis or the vas deferens. All right. And it would be really great if you could, but that's part of the reason why you have to have conscious male going through an ejaculatory cycle to get viable sperm. Then usually when they get the sperm, they wash them, they remove extraneous, they freeze them, and then you go to a sperm bank and you buy the frozen sperm to potentially use if you're going to use a sperm donor for fertilization. So every viable sperm usually comes from an ejaculation. All right. Okay. Again, your epididymis, head, body, and tail, not head, neck, and tail. The neck goes with the sperm, all right? And again, it's long. It's 7 meters, 23 feet of tubing, okay? And in the epididymis, you recycle damaged spermatozoa, you store and protect spermatozoa, you put a lot of secretions in there that continue to push the maturation and the cell cycle of the sperm to become realized in a few days to weeks a swimming sperm that is capable of fertilizing an egg. Okay, so head, body, and then the tail is down here. And again, the tail is the target in a when the male goes to get a vasectomy, is the target to try to cut to make no more connections between the epididymis and the vas deferens. If some male decides I want to try to reconnect, it is possible that if they can find the vas deferens or the ductus deferens and try to reconnect it, there could be a chance that sperm can still get through. There's also a chance that they could cut it, and if they don't kind of cut enough and let it separate away, that this could still be close enough that sperm could get through. So while a vasectomy is very, very effective, there is still a chance it could not work. And there's also still a chance that for a vasectomy, if you do cut away and break them apart, you could still bring them back together and then return fertilization capability or sperm making capability later. So in a vasectomy, someone who then ejaculates sperm, the goal would be the sperm is so few to zil, there's no capability of then fertilizing an egg because you're not delivering any sperm, or very few low numbers of sperm, so low that the odds or statistical ability to hit the egg and get to the egg would be very, very low. But it still can happen. Okay. All right, the ductus deferens is, again, a long tube. It is going to begin at that tail of the epididymis. It's going to have to go up around the bladder and then connect in the back where it enlarges, and think of that as like the little holding enlargement where the sperm collect. That's known as the ampulla. The ampulla is next to the seminal vesicle or the seminal gland that is basically kind of sitting like two cars at a stop sign. They're sitting in parallel, and they're going to produce their secretions, the sperm and the fluid, into the ejaculatory duct. That's where that combines. Prostate adds in its little sprinkling of fluid and influence, and then everything exits through that ejaculatory duct into the urethra. Okay? All right, so again, here's the ampullas. Here's, again, that coming over. This is the bladder. The seminal vesicles or the seminal glands are going to be right next to the ampulla, and then everything is shooting into the ejaculatory duct, and then the prostate will push in its secretions into that ejaculatory duct. So what ends up in the prostate urethra will be sperm and secretions from the prostate and secretions from the seminal glands, and that will start triggering the sperm to awaken, to become more metabolically active. So when they are ejaculated very shortly into the female, we start to see the sperm have capability to swim and move, and with other items in that ejaculate that cause smooth muscle contractions of the uterus, throw those sperm as close as possible to those ovarian tubes or fallopian tubes. So some of the sperm going right and some of the sperm going left are swimming up in those tubes towards hopefully an egg to fertilize. Okay. All right. And again, your urethra, you have the prostate part. You have that little area where, again, the bulbourethral gland, the pelvic floor would be, the muscles that kind of represent where your body, your abdominal pelvic gland cavity is ending, your pelvic floor, and then the external genitalia is starting. That's the membraneous, and then the spongy. Okay, just a different picture. And again, the goal of these glands are to the bulbourethra, the Cowper's gland, preemptively lubricate, preemptively cleanse the urethra. The other two support and provide the right constituency and fluid and pH balance to get the sperm awakened, ready, and activated, and to help promote the sperm towards the to the egg. Okay. So again, your seminal fluid is slightly alkaline and has prostaglandins that push smooth muscle to contract. Remember prostaglandin, when we had a clot, it was trying to get contraction of the vessel that was damaged, so we stopped blood flow through there. Well, in the uterine case, it's trying to contract the uterus, so sperm are getting kind of like a trampoline thrown up. And as they get thrown up, hopefully some of them shoot in to the uterine tubes. All right. Fibrinogen is going to help, again, kind of temporarily clot the vagina. So hopefully stuff in the vagina and in the uterus start to, again, have higher odds of sticking in there, staying in there, and getting towards the egg. Okay. Fructose, remember glucose becomes glucose 6-phosphate, fructose 6-phosphate, 1,6 by fructose. So fructose is part of the breakdown. It's a fuel source. It's one of our monosaccharides that we make ATP from. Okay. Again, your prostate gland is going to add in a little bit of acid, so the acid and the base can kind of equal out, and you don't get too acidic or too alkalytic from just one. They kind of work together to make that proper amount of acid and base, so our pH is a little bit more basic than normal, but not too basic, and the sperm are not killed off for that. All right, the bulbourethral gland is going to be very alkaline, and that's preemptive. So the acidity from the prostate also somewhat helps kind of, I have base and base, it kind of modulates so I don't get overwhelmed with too much base. Okay, and it's going to give me a little bit of some antibiotic molecules, again, to protect the sperm from foreign entities and to somewhat protect the sperm from being attacked by the female. Okay, and it's going to trigger some molecules to trigger peristalsis and movement of the sperm out. Okay. All right, bulbourethral gland is the other one. Again, it's going to be helping with lubrication. There are going to be also glands in the penis external genitalia area, and it's part of those penile glands that we tend to see the milkier, waxier external secretions that sometimes we associate with people that still have foreskin as being unclean. Okay, and again, your semen can be from anywhere from 2 to 5 milliliters, and in each milliliter, there can be somewhere between 20 to 100 million spermatozoa. So if you do the math, if I'm on the low end and I have 2 mils, and in each mil I only have 20 million sperm, my entire ejaculate is maybe 40 million sperm. If you're on the high end, which many of you younger teenage boys might be, if you get 5 mils, and let's say in that 5 mils I have 100 million spermatozoa, I'm looking at 500 million sperm in that ejaculate. So that's part of the spectrum of, I can be anywhere from 40 million to 500 million sperm in this 2 to 5 million potential ejaculate. Right? And that inhibin FSH Sertoli cell is trying to keep, again, your range of, I make enough sperm that I'm making 20 million sperm per milliliter to 100 million. I'm not over or under producing. Okay? Again, the penis is going to have a root, a shaft, and then the glands, or the distal part, is going to be surrounded by some skin that's going to have, again, some external glands that are going to help with lubrication. Right? Inside the penis, I'm going to have 2 big blood vessels surrounded by a open or meshwork of spongy material known as the corpus cavernosum. That is the main erectile or most robust erectile tissue. And then around the urethra, I'll have another piece of erectile tissue known as the spongiosum. Okay? Now, the key to the erection is the blood vessels need to dilate. So I need less sympathetic outflow, less norepinephrine, less prostaglandin, less stress, less high blood pressure, and I need the ability of these vessels to dilate and the blood amount, and therefore the local hydrostatic capillary pressure to go up. So then the blood engorges these vessels, more fluid is leaked out, more fluid engorges the corpus cavernosum, the corpus spongiosum, and that's how I get an erection. So one of the big reasons why people with high blood pressure can't get an erection is these vessels and these bloods in this area, it's already maxed out at a high pressure. It can't go up anymore, and they can't get then the pressure high enough to force that fluid into the spongiosum and corpus cavernosum to get an erection. So that's why if you can get them to, with like Viagra, dilate those vessels and that local area becomes engorged in more fluid, and I can get more blood pressure when I normally am sitting at a high blood pressure already, I can then get that erection. Okay. All right, again, the dermis of the penis has more dartos muscle. There's some underlying areolar tissue, and there's lots of, again, arteries, veins, lymphatic vessels, and lots of sensory vessels, sensory organs tied to the nervous system. Okay, the foreskin is known as the prepuce. It has these glands which secrete somewhat of a waxier, thicker material, and that's part of, again, some of these modified glands. All right, and back in the day, again, people didn't bathe regularly, people didn't clean, and you will probably see people who have obesity and these folds, and when you lift up some of the folds, the sweat and the goo and the grime collects. So imagine that in this foreskin area. And so circumcision, and it was practiced again as a cleanliness thing, and a more sanitary thing, but circumcision is normally done in Western culture to help, again, remove that excess skin so you don't end up with potential, like an obese person and their rolls, the accumulation in these folds of dirt, grime, sweat, and this waxy material. Circumcision is not practiced everywhere. In fact, culturally, in my area of Louisiana, my grandparents and probably even some of the older parents of my parents' generation, they were not always circumcised. You didn't always go to a hospital to have your baby, so you didn't always have a doctor to circumcise, and we were Catholic, not Jewish, so it wasn't a big event of birth or religious significance to be circumcised. So circumcision is usually going to be happen by your it doesn't get done by the pediatrician, it gets done by the OB. So circumcision is something, if you go into a Western hospital in Europe or in the United States, you usually have to tell your OB you want the baby circumcised, and they will do it before you leave the hospital. If you have a home birth, you might have to make an appointment with a pediatrician or your OB to then circumcise the baby. Okay. Nowadays, there's a big push culturally for kids to choose to be circumcised or not. Circumcision is something that the longer you leave it, the more likely it's going to be painful, because you've made those, neural connections to your sensory system. And you are more likely as you get older to also have erections and morning, dreams and things that can trigger erections, and then you can stretch the skin when it's trying to heal and cause damage. Just go watch Shameless. There was an episode of Shameless where Carl gets circumcised, and he's becoming a sexually active, like 13, 14, 15-year-old, and it becomes a problem because he keeps damaging the healing skin because he keeps getting erections. All right. Again, we already hit that. Corpus cavernosum, corpus spongiosum, and again, your hormones, we hit that already. All right, I'm going to stop here, and we'll pick up with the effects of testosterone